observations from lab scale systems related to bulking sludge formation. And these lab scale systems are all scaled down versions of full scale reactors with particular substrates and with a particular operation. The example here, we use a cycle length of 4 hours, which means we have an hydraulic retention time in these systems of 8 hours. We applied a solid retention time for as far as possible of about 10 days, which is not always possible if you get bulking sludge, because then the settling is not good enough. And we used acetic acid as a kind of resembling the easy biodegradable substrate in influent. If you look to wastewater composition, then from the soluble COD, a large fraction is usually acetic acid. We can now change the length of the feed phase, and by changing this length of the feeding phase, we change effectively the behavior of a reactor. If you have a very short feeding phase, it's like a plug flow reactor. If we increase the feeding phase length, it's like having a selector, and if we would feed through the whole cycle, all the 4 hours, it would mean we have a continuous, completely mixed type of reactor. And we can of course vary the oxygen concentration there. So between micro-aerophilic and very well aerated systems. Now what do we see? There's a lot of information on this sheet, maybe too much, but in the graph on the right upper hand, you will see the experimental time, which is the time after we started the reactor with normal, well settling sludge with a volume index of around 100. And the different lines are for different lengths of feeding periods. So 3 minutes is a plug flow reactor, 193 minutes is effectively a completely mixed reactor, and the longer the feeding period, the more, say, completely mixed the reactor effectively is. Or you could say the larger the selector would be. And what you see that if you have relatively short feeding periods, so like in a selector and like in a plug flow reactor, you get a stable sludge volume index, which might even slightly decrease. Whereas if you have longer periods, even of 20 minutes, 60 minutes, or not 90 minutes, you see that the sludge volume index in time rapidly increases, and the more completely mixed the tank is, the more the competitive advantage obviously is for filamentous bacteria, the quicker the sludge volume index is increasing. So what you see is that the shorter the feeding period, the better the sludge volume index is. The longer the feeding period, the more mixed the reactor is, and the more the filamentous bacteria get the advantage, and the more rapid they grow in, the more quick the sludge volume index increases. And of course, if it gets above 300 or 400 milliliter per gram, it's not possible to maintain the reactor in proper conditions, because all the time your biomass is washing out. Now if you plot that in a different way in the graph left below, then these data are plotted as the feed length over the total cycle length. So it's the period of the time the bacteria see substrate effectively. So if it's 2%, then 2% of the total time they are in the reactor, they really see substrate, and the rest of the time they are starving. And in this time, they take up the substrate, they store it, and the rest of the time they grow on the accumulated substrate, which is accumulated inside the cells as fat reserve effectively. You see that if you increase that feed length period, so which is the same as, say, increasing a selector, which is upfront a completely mixed tank, you will see that initially there's not much effect on the volume index, but if you go, say, from 8 to 12% of the volume of the feed length, you will see a rapid increase in the sludge volume index. And you also, the yellow line displays something different. That's the rate at which the bacteria take up the substrate as a relative rate. So if it's 1, they take it up at their maximum rate. If it's 0.4, they take it up at 40% of their maximum rate. Now what you see here again is that if you get more and more in the direction of completely mixed, filamentous bacteria get the advantage. They will take over. But what's even more important, that it's a very small margin. Because if you would say I build a selector tank which is 8% of the volume of the total system, or a selector tank which is 10% of the volume of the total system, will give a very big difference in sludge volume index, which you in the end get. And 8 or 10% of the total volume, that's just 2%, and it's very small. Predicting the flow rate towards the wastewater treatment plant by 2% accuracy, because that's what you effectively would require if you would like to do it in this way, is not very easy. So this also points to the fact why it's so difficult to properly design selectors, and why in practice you often see that people build a selector in one plant and they think, oh, we do the same in the next plant, without realizing how to properly design it. They just copy, and then it doesn't work, and there it works, and there it doesn't work. And that is because this very sensitive influence of the size of the, say, the feed period relative to the total period on the bulking properties of the sludge, on the advantage the filamentous bacteria get. It's a very critical design parameter, and that's also why you get these problems in practice with a lot of different stories. And of course this 2% variation in volume, yeah, that's almost impossible to really measure properly, even in a wastewater treatment plant, because measuring, at least in the Netherlands, influent flow rate measurements have typically already 5 to 10% standard deviation, and then we need 2%. So what you see is increasing this length of the feast phase, which means lower substrate concentrations in the reactor, which means that we get a deterioration of the sludge settleability. So clear relation between substrate presence and the substrate uptake rate, substrate concentration, and advantage for filamentous bacteria, which fits with the hypothesis of the kinetic selection theory, which I described before. We now looked to, in the previous it was effectively looking to 2 large selectors, but you can also have a 2 small selector. So what you can do in a sequencing batch reactor, you feed 100% of the, or say 80% you feed at the start of the cycle, you wait in half an hour, and the rest of the cycle you feed the rest of the 20% of the substrate. That's like if you have a selector system which you build too small. Part is taken up at a high substrate concentration, but at the end of the selector not all the easy biodegradable substrate is consumed, and that ends up then in this completely mixed reactor system. Now if you look there, then we see that if 10% is not taken up in the selector, so the selector is effectively 10% too small, then that's not really a big problem. But as soon as it's 20 or 30%, it is a problem, because we get again within 2 weeks bulking sludge conditions. And again pointing to the fact that if you want to design this kind of aerobic selector systems, you have to design them very accurate. With say 10% inaccuracy is allowed, but 20% of volume not. And again, if you realize how much hydraulic flow rates vary in a wastewater treatment plant, you might realize that it's not that easy to build with this accuracy a selector for a wastewater treatment plant. So that's for aerobic selectors. Then we can also look to the effect of aeration. And we can look to the effect of the oxygen concentration. And what we typically see there, that if we decrease the aeration in the selector, so it's not the oxygen concentration in the main tank, that we all the time keep high, so there's no limitation in the, say, what we would call normally the completely mixed tank, but during the substrate supply, during the time that they take up the easy biodegradable COD, we regulate the oxygen concentration, and we can regulate that at 0.2 milligram per liter, at 1 milligram per liter, at 2 milligram per liter, and we see that the lower we make the substrate concentration, that if you go below around 1 to 2 milligram per liter, we will get all the time again bulky sludge formation, and the lower the substrate concentration, the earlier it occurs. So effectively for oxygen, it's the same as for the soluble for the substrate. If the bacteria take up the oxygen at a rate which is not their maximum rate, because the oxygen concentration is too low, if the oxygen concentration decreases, uptake rate decreases. So if the bacteria take up the substrate at a rate which is low, or at a low substrate concentration, we get preference for filamentous bacteria, in accordance with the kinetic selection theory. And for the bacteria, effectively, oxygen or acetic acid, oxygen or glucose are the same thing. For them it's two chemicals they need, and they need to take up. And so it also doesn't matter whether the oxygen concentration is low or whether the substrate concentration is low. In both cases, you will get bulking sludge. Now you also see here again a problem for selector design in practice, because people typically measure the oxygen concentration in the main treatment plant, not in the selector. And if you ask people, how is your oxygen concentration, they will say, oh, it's all the time high, it's sufficient. But the selector, if you have an aerobic selector, about all your COD, easy biodegradable COD, is removed in the selector at a very high rate, which means you have to also supply oxygen at a very high rate. And that's not always possible. So if you have an aerobic selector and you don't add enough aeration, then the selector again will not work. And in practice, what quite often happens is that initially the selector works, but then in time the aerators fail, or they become not that efficient anymore, aeration efficiency goes down, oxygen concentration in the selector goes down, and we get bulking sludge again. So it's again one of these problems in practice. If a selector doesn't work, you have to really evaluate why is it not working properly. Is it too small? Is it too big? Is the aeration not good? Different options which can happen and which you would have to investigate in detail in order to evaluate. Here in the lab scale experiments we can show very reproducibly that we all the time will get bulking if the oxygen concentration is too low. Now again, effective aeration. If you look to, it becomes a little bit more complex. What we did here again is experiments with different aeration intensities in the selector. We can have high aeration intensities, the green and the white curves, then no problem. We all the time get very nice settling sludge. If we have lower aeration intensities, the lower the aeration intensity, the more easily we get bulking sludge. Once we have bulking sludge, like in the red and the yellow line, we can increase the aeration intensity, and we see that the sludge properties go down again. So the flock-forming bacteria can gain the competition again, so we can take this remedial action. After getting good sludge properties again, we can go back to the old oxygen concentration of 0.7 or 0.2 milligram per liter, and we see a rapidly increase in filamentous bacteria again. And so it's reversible, we can reproduce it. It's directly the oxygen concentration at which the bacteria take up the substrate which is important. What you also see is that the responses are slow. We take on day 6, we take the measurement to change the oxygen concentration in the tank, and it takes, well, about 10, say almost 2 weeks before the sludge comes back to a normal state. So we take an action and there's a response time of about 2 weeks. It's a slow response, which makes also that, and this is in a nice laboratory system where everything is controlled, where we know what we're doing. In practice, the response will be even slower in full-scale treatment plants. So this is another problem which you have in interpreting what's going on with bulking sludge. We're dealing with microbial competition effects, and it takes 1 or 2 SRTs, solid retention times, before we fully get the effect of the control actions we took. And that makes it again difficult to evaluate, because in the lab we know what we're doing. In a full-scale treatment plant you change one thing, but usually 2 or 3 other things are also changing. Temperature is changing, it's rainfall period, it's there's some maintenance of a pump or what else. There's usually more things to do. So correlation between action and response is not that easy. On the right hand side you see also a figure again of an experimental time. But now we look to prolonged feeding, and again with different oxygen concentrations. And then again you see that the low oxygen concentrations lead to bulking sludge, but not that quick anymore as when we give pulse feeding. So what we see that, well, first these experiments are very reproducible, so the behavior of the bacteria towards low oxygen concentrations or towards short or long-term feeding, so low substrate concentrations, is very reproducible. Low concentrations all the time lead to bulking sludge. Oxygen concentration has more or less the strongest impact. If oxygen concentrations get lower, it has more stronger impact even than the substrate concentrations. But what's interesting to see, if both are low, so if you have a completely mixed tank reactor with a low oxygen concentration, then you get less bulking than when you have only a low oxygen concentration or only a low substrate concentration. And so if both substrates are limiting, the net effect is less than one of the 2 concentrations is limiting. This again points to the complexity of bulking sludge, because you have to evaluate all parameters. If you only look to oxygen, then in one case, if you look to the left graph, the yellow line, you see within 5-6 days increase in sludge volume index due to the low oxygen concentration. In the right line it takes a month to reach more or less the similar sludge volume index. So the response at the low substrate concentration is much slower. So you cannot only evaluate only oxygen, you have to evaluate the oxygen and the substrate and potentially other nutrients. That makes why it's again complicated if you have full-scale systems to get data out of there and to really analyze what's going on. At the lab, of course, if everything is well, you know what you're doing. What you also see is that at the different concentrations, oxygen concentrations, we all the time found different filamentous bacteria. And here the question is, why are they different? But that's maybe partly, mainly a very interesting, by the way, academic question, because in practice, if your oxygen concentration is low, it doesn't matter which organism you get. It's too low and you cannot operate your treatment plant well. You just have to make sure the aeration is proper. So if you look to this bulking sludge, this kinetic selection theory, then we see if we use the theory and we just use it in general terms, then we will find that we can explain our observations pretty well with the theory or the hypothesis brought forward by Chudaba about selection, low substrate concentration filaments, high substrate concentration flock formers. So if you want to use it for explaining what's going on, it's a very proper theory. However, if we look to the sludge we have and we measure on the bacteria the maximum rates and the maximum and the Ks values, then we see that for the filamentous sludge as well as for the flock forming sludge, we find the same maximum rates. We find also the same pHb storage rates, and we find also similar Ks values. They are more difficult to establish, but they are also in the same range. So we measure that the basic hypothesis of this theory is not correct. So that is a little bit a problem, because you have a nice theory or a hypothesis, it works, you can really use it to optimize your system, no problem with that, but the mechanistical explanation which is below it seems not to be correct, because the filamentous bacteria you cultivate under these conditions have just the same maximum substrate uptake rate as the flock forming bacteria, and the basic assumption was that they are different. So I will come back to that later. what we effectively see, if we now make a kind of listing, then we see that substrate concentration at which the bacteria take up the substrate, so effectively which sets their growth rate, is very important for the type of morphology we get. If the substrate uptake occurs at 80% or more of the maximum substrate uptake rate, then we get very nice flux and even formation of granules. And we see very few filamentous bacteria. And we get very nice granular particles in the reactors. If we go to somewhere between 60 and 80% of the maximum uptake rate, so we dose the substrate at a rate that the bacteria can take it up at 60 to 80% of the rate they can maximally achieve, then we see, let's say, the regular activated sludge flux, porous, open structures, also again a few filaments, some finger-type structures. However, if we now go to substrate uptake rates at, say, 50% of the maximum rate and lower, then we get dominance of filamentous bacteria. We end up with bulking sludge in the system. So it's again quite narrow ranges effectively if you think about that within process design. But if the substrate concentration decreases at which the substrate is taken up, you just will get more filamentous bacteria. That's the observation. And we have two things. We have the substrate and the oxygen. And so we can define more or less areas where at low dissolved oxygen we will anyway get bulking sludge. At a low substrate uptake rate we get bulking sludge. And there's areas where we can get very well settling sludge if we make sure that the oxygen concentration is about 1 to 1.5 milligram per liter and we have a high substrate concentration. If we have high oxygen concentration and an intermediate substrate concentration, we get, say, light sludge, which is still normal sludge for practical activated sludge systems. And this is all, of course, for conditions where we have easy biodegradable carbon, like acetate or glucose. We have aerobic conditions and no nitrification, denitrification, because that will all furthermore complicate the system. This is just for a COD, easy biodegradable COD removal system, which you will quite often find in the industry and in wastewater systems where the sewer is anaerobic, so there's a lot of easy biodegradable substrate present in the wastewater. But we can also look, of course, to denitrification in these lab systems. Again, an SBR, but now we have an anoxic aerobic reactor where we have the substrate addition under denitrifying conditions. Then, after the denitrification period, we start aerating. Then we can settle and we can draw. And again, we operate in this case cycles of about 6 hours, hydraulic retention time of about 12 hours, and a solid retention time of about 10 days. So, in the range of nutrient removal processes. And what we see, without going into all the details, that if the anoxic selector is too big, we get a low substrate concentration, again, at the uptake, and we can also simulate a low nitrate. Similar nitrate replaces oxygen, so we can also look to low nitrate concentrations. And what we see that if we have pure denitrifying conditions, the conditions which normally would lead to bulking sludge under aerobic conditions don't lead to bulking sludge under denitrifying conditions. So, not a low nitrate, not a low substrate concentration, both will not give any bulking sludge. So as long as it's purely denitrifying conditions, there seems not to be able to form bulking sludge. And also in the literature, by the way, there are no filamentous bacteria described that can denitrify. This does not mean that they don't exist, because there's no reason why a filamentous bacteria cannot denitrify, but under the conditions studied with acetate as carbon source and 20 degrees Celsius, they don't appear. And that they don't appear, it's also interesting to see at least that with although we have these extensive studies, there's no denitrifying filamentous bacteria described. In laboratory and pilot systems, there are often not pure denitrifying conditions. And that gives the problem. If you have a lab scale reactor and you have just the denitrification tank open to the air, you effectively have microaerophilic conditions because oxygen penetrates through the headspace. And even in sometimes selector systems are only a few 1 meter deep or something like that, or you have overflow weirs, etc. You measure an oxygen concentration of 0 in the reactor, but due to some headspace aeration or aerated sludge recycle, you still have some oxygen present in an anoxic selector, or you might have, and then you don't have pure anoxic conditions anymore. You effectively have microaerophilic conditions, which indeed lead to bulking sludge. For anaerobic selectors, that's effectively what you do if you have a BioP system also. You can make an anaerobic selector. And in the anaerobic selector also the easy biodegradable substrate can be taken up. In this case, not by normal heterotrophic bacteria, which use oxygen or nitrate as an electron acceptor for their growth, but they will be taken up by bacteria like phosphate accumulating organisms or glycogen accumulating organisms, bacteria which found ways to generate energy, limited amount of energy, and use that energy only to take up the substrate. They cannot grow under these anaerobic conditions, but they can take up the substrate, and if then later on in the system they see oxygen or they see nitrate, they can start growing on the substrate taken up. But effectively in this way they also remove the substrate from the wastewater. Now, again, we can look to if you have an anaerobic selector, whether it's too big or too small. Under similar conditions as we worked for this denitrification. Now with the anaerobic selector we see that we always get very well settling sludge, much better than under the denitrifying conditions. Sludge volume indexes of 40 milliliter per gram. That's effectively granular sludge. You also see that again, here there's no influence of the substrate concentration. We can feed all the time, they can take up the substrate at a few percent of their maximum rate. It will not lead to a deterioration of the sludge. So obviously again, you would assume that there are no filamentous type of organisms which can have this kind of physiology. Moreover, what we also saw that if we make the anaerobic selector too short, so that we have spillover of substrate into the aerated phase, the system is less sensitive to it. We could spill up to 30-40% into the aerobic reactor, into the aerated space, so make it a much too small anaerobic selector, maybe 70% of the needed volume, and still get good sludge properties. So the design of the anaerobic selector also proved to be less sensitive to the bulking formation, which is of course also important. Now this is all for soluble substrates, but we also can have particulate substrates. And particulate substrates, they need hydrolysis. If you have, for instance, starch, the substrate we used here, starch has to be hydrolyzed into maltose, and the maltose is taken up by the bacteria. Now the problem here is, if you think about it, that the hydrolysis of the starch is a slow process. So as soon as the maltose is released from the starch, it's taken up, meaning the maltose is all the time taken up at a low substrate concentration. In accordance to the kinetic selection theory, and what I discussed before, you would expect that if you have maltose taken up at a low substrate concentration under aerobic conditions, you will get filamentous bacteria growing, and you will get bulking sludge. So it's interesting to add then the starch, and you can add two types of starch, soluble starch and particulate starch. Now the soluble starch will for a large part end up in the liquid and absorb to the flux and slowly be hydrolyzed. the particulate starch will very rapidly be incorporated inside sludge flocks and also be hydrolyzed. So they have a little slightly different behavior. We give pulse feeding. It doesn't matter because the starch is not the direct substrate, it's the maltose. The maltose is produced during the whole cycle length. So it's bacteria all the cycles see maltose and take it up at a low concentration. And what we see then, that's on the left hand side, that's a nice thing of using starch. It reacts blue if you add some iodine. So on the left side you see that the most of the starch will, as long as there is starch present. What we also find is that we indeed, in accordance with the kinetic selection theory, we get filamentous bacteria, Nosticoida seemingly. But despite the fact that we get a complete dominance of filamentous bacteria, we don't get bulking sludge. And also this has been reported moreover in the literature. There's no direct relation between the growth of filamentous bacteria and occurrence of high sludge volume indexes. So occurrence of high sludge volume index or bulking sludge. Here you see a filamentous bacterium on this picture, which forms a nice and compact sludge flux with a volume index of less than 100 milliliter per gram. So it points to the fact that this kinetic selection theory might indeed be correct, that indeed filamentous bacteria win the competition from normal bacteria if the sludge concentrations are low. But maybe when you get the theory, when you get bulking sludge, when you get an open and porous flock structure, has some other background or some other mechanism. And what is different here is that in this case, the substrate originates from inside the flock and not from outside the flock. Normally the maltose would come from outside the flock and being degraded at a low concentration, meaning that the substrate concentration outside the flock is high and inside the flock is low. That's the green line. If you have a substrate which is hydrolyzed, it's the other way around. The maltose concentration inside the flock will be higher than outside the flock because it's produced inside the flock. So the bacteria grow in a different substrate gradient. They grow still at low substrate concentrations, but the gradient is different. So maybe the substrate gradient has something to do with whether you get bulking sludge or not. Because if you would give maltose at a low substrate concentration in the feed all the time, then you will get bulking sludge. Now you see also that the substrate which is more effectively taken up inside the flocks, the native starch, gives a better volume index than the substrate which is soluble and which will be mainly hydrolyzed at the outside of the flocks. Because the soluble starch will absorb partly on the surface of the flocks and there being degraded, whereas the particulate material will just absorb and be integrated in the flock structure. And that might again lead to differences in the gradients of maltose which you observe in these flock structures. So that's what we learned from using a particulate substrate. We see again that the kinetic selection theory might hold indeed for selecting filamentous bacteria, but that we might have to make a distinction between selecting for filamentous bacteria and selecting for filamentous flocks, because they could be different. The last organism which is giving presently the most problems, at least here in Western Europe, because it's associated to the occurrence of to the low-loaded activated sludge systems, nutrient removal systems particular. So, and that is the organism called Microtrix parvicella. It's one of the most studied filamentous bacteria. Actually only 1 or 2 people have ever managed to grow them in a pure culture. So again, most of the knowledge comes from observations on sludge. And we also try to grow that into our system. And now I would have to explain a carousel process. I'm not sure whether you all are aware. It will have been handled in a different lecture. But a carousel type of process, or an oxidation ditch type of process, you have one point where there's a surface aerator in general, and then you have a long loop where the sludge mass goes around, and every, say, 10 minutes it passes by the surface aerator. So every 10 minutes it gets aerated, and then it goes around, and halfway oxygen is gone, and you get denitrifying condition, and in this way you have simultaneous nitrification, denitrification inside the treatment tank. We can also simulate that in a sequencing batch reactor in the lab by all the time giving short pulses of aeration. So like in the tank, every 10 minutes the sludge gets aerated for 1 minute, and then it travels through the tank. So we can do that in the sequencing batch reactor by every 10 minutes giving 1 minute of aeration, 10 minutes not aeration, 1 minute aeration, 10 minutes not aeration, and that for the whole length of the process. It's like going around in the reactor, and that's what the sludge flock experience. Now we know the substrate for microtricks are lipids. So we give a substrate which contains mainly lipids, called tween 80, normal growth medium for bacteria. A particular N source, a particular sulfur source. The bacteria cannot grow on sulfate. They need methionine for their cell synthesis. And we make a DO control, which is in such a way that in the end we have 30% aerobic mass fraction, which is similar to carousel and oxidation ditch systems. And under these conditions you indeed can accumulate this organism in a lab scale reactor. So you can again see that a bacterium which is very difficult to grow in pure culture, if you analyze what the organism experiences in a real system, and if you analyze what its real substrate is, you are able to cultivate it inside a lab system, not in pure culture, but you can accumulate it and you can study it. And so obviously this organism somehow benefits from the advantage that it gets all the time these short oxygen pulses and these low oxygen concentrations. So if we look to why does bulking sludge occur? What have we learned from all experiments, literature, etc.? Then we see it's a combination. It's a combination of a certain microbial morphology and gradients of substrate concentration in sludge flocks due to diffusion, reaction and growth. The substrate has to diffuse from outside into the sludge flock, and that means that at the outside you have a high concentration, inside you have a low concentration, very similar to biofilm systems, which are handled in a different lecture. And that means that also a bacterium which sits at the outside of the sludge flock will observe a higher concentration than a bacterium which sits at the inside of the sludge flock. So the bacterium which sits at the outside will grow faster than a bacterium which sits at the inside. So the diffusion gradient will give advantage to the bacteria which grow at the outside of the flock. And it might well be that that leads to open and porous flock morphology rather than the fact that the bacteria are present as such. And we saw with the grow on starch that it's very possible to get filamentous dominated system but still a very compact sludge flock. And just to show a little bit what's feasible also in modeling nowadays, this is just an example of a model which is very similar to a biofilm model, where we treat bacteria as individual organisms. We describe each bacteria individually, and we can have two different types of bacteria, one which makes flocks and one which makes filaments. And as soon as you make models, you already have to think about and you realize what the real difference is. Because a flock-forming bacteria, if it divides, one cell will be positioned somewhere around its mother cell. You will get a globular growth. If you have a filamentous bacterium and it grows, the next bacterium will put in the same line, which means that it grows in only one dimension. If it has the same growth rate, the speed at which it widens is much more faster than when it grows as a globule. So the filamentous organism will tend to grow much more faster out of a sludge flock than a normal bacterium, which would, say, fill up the total volume. Now we can describe that, we can make models which take this morphology into account and describe that with just the normal reaction and diffusion processes, as also are dealt with in the mass transfer lectures and in the biofilm lectures. We can do that in 3D. And what you see is, this is just to show you a little bit how it looks like. If you have flock formers, they form a globule. If you have filaments, they grow in one direction more or less. And you can add, because bacteria are sometimes bend or branch, you can add, of course, that feature in a simulation model. Now you can also add attachment, because that's one of the other aspects. You see, if you have filamentous bacteria, then such a filamentous organism can function as an attachment site for a flock-forming bacterium. That's what you can also clearly observe if you look under the microscope. And these flock-forming organisms, they start to colonize the filamentous organism. If they would get a chance to grow out, they would fill up everything and you will have no bulking sludge. And of course you need some detachment because cells leave the flock again. Now if you do that, and you now take only flock-forming bacteria, so no filamentous bacteria in the system, then we'll see that at a low DO concentration the flock grows slower. It takes longer to grow it because bacteria grow slower. But also, on average, the structure is more porous and more open than at the high dissolved oxygen concentration. So already if you have the same morphology, the porosity of the system increases at a lower oxygen concentration, which shows that a lower oxygen concentration leads to a more porous and open structure, independent of the morphology of the bacterium itself. And that's also the same for biofilm systems. And that's due to these gradients. Now if you now have include the filamentous bacteria in such a simulation, you do the same simulation again, but now with filamentous bacteria, then you see that these filamentous bacteria, of course, because they only grow in one direction, they very rapidly grow out of such a flock system, and you get attachment and growth and new colony formations at different parts of these filamentous organisms, but you get a very open and compact, a very open structure, where in this extreme case you have filaments with here and there some flocks attached, flock-forming bacteria attached. And in this case, we used exactly the same kinetic parameters. So we used the same growth rate and the same Ks value. But effectively, because the flock tends to be more open and porous, an organism which grows more open and porous gets then also the advantage. So the fact that you get these filamentous sludge flock formation is more the result of the gradients. The fact that you get filamentous bacteria might indeed be due to this kinetic selection theory. Although the growth rates don't need to be different, then at least the Ks values might be different. And this is just a picture to show for such a sludge flock. The dark gray, the big black contained area, that's a sludge flock. The grayness is a measure for the concentration, and the arrows are a measure for the speed at which the substrate diffuses towards the flock. And you will see that at the tips you have much higher speeds of substrate transport, so also higher substrate consumption. That's also why these tips grow out more fast. And if then at the tip you have a filamentous bacterium, it will grow out even faster because it will grow out more or less only in one direction. So which is then most likely also the reason. So if you think about it, then why do you get bulky sludge? It's morphology of the cells plus this gradient of substrate concentration in the sludge flocks. That's why you get bulking sludge. And you can get filamentous bacteria because of low substrate concentrations, as we saw with starch. And they can indeed compete out probably at a low substrate concentration. but that's not the reason to get bulking sludge. The gradient is an anti-bulking sludge. Now, in this context, it's not a problem if you take the kinetic selection theory, because it's a little bit easier to understand than this reaction diffusion mechanisms, for explaining what's going on. Because if you use it and use it properly, the remedial action you take within the wastewater treatment plant will anyway go in the right direction. So for that point, it's a very strong theory to use.